Distinct pleasure to introduce Dr. Elhanan Borenstein. He's currently the Associate Professor of Genome Sciences at University of Washington, just in Seattle. He actually was gracious enough to drive up, and he's going to drive down just for tonight, just for a day trip. So it's really nice to have him here. He obtained his PhD at Tel Aviv University in Israel in computer science, and prior to that, he was telling me that he actually was involved in the high-tech industry and tried to and did start a company and tried to develop softwares for what is it? Develop software for something that's no longer of his interest right now. So that's why he went and did his PhD in computer science, and he did a postdoc at Santa Fe Institute and at Stanford University, and he's still affiliated with Santa Fe Institute as external professor there. His research interest is in metagenomics and integration of metagenomics data using various techniques that he will tell us about today, and with the goal of trying to understand the microbiome and how it can affect human health. He has won quite a few awards, including the Alfred Sloan Fellowship and also the NIH New Innovator Award.
So, hi everybody, and good evening. It is an absolute pleasure to be here and to tell you a bit about the work that I do in my lab, specifically in trying to develop methods and models that use a system biology approach to analyze metagenomic data. Before I do that, I really want to thank Will, first of all, for this very kind introduction that leave me no choice but to disappoint you, of course. But more than that, for inviting me to speak here. And of course, working in bioinformatics in Seattle, you know that there is this big bioinformatics community just 3 hours, or actually 2.5 hours, as I discovered today, away from you. But of course, coming here and talking to some of you and talking to the students and seeing this room full of people help you to realize just how active and how vibrant this community is, and I'm really happy to be here. In fact, when I was preparing for this visit and I trying to get a sense of what I actually know about bioinformatics in Vancouver and bioinformatics in Canada, I realized I have a good testament for that in my own lab, because it turned out that out of the 5 postdocs that I had in my lab over the last 3 years, 2.5 or 3 were actually Canadian. And so if you do the contingency table, you have an extremely strong p-value for just the quality and how wonderful this community is. And first of all, I just wanted to share this with you, but also my goal in life now is to get the p-value stronger. And so if there are people here that are interested in computational postdoc in Seattle, definitely talk to me. Having said that, what I will try to do today is to tell you just a bit about the human microbiome, and I want to thank Thea for giving this great introduction and saving me the time of presenting the idea of metagenomic. I will say a few things about that, but I will mostly try to convince you why some of the things that we do in microbiome research now are somewhat limited, and specifically why we need some of the approaches that are borrowed from system biology. And the majority of the talk I will invest in reviewing multiple projects from our lab. And again, in preparing for this talk, I was kind of going back and forth between, do I want to talk about one project and give you all the nitty-gritty details, or give you a more broad overview of my lab? And because I do see this visit potentially not just as a one-time visit, but as a way to promote more dialogue and more collaborations, I really wanted to share with you a lot of different projects as a way to get you familiar with the kind of thing that we're interested in, the kind of thing that we hope we know how to do, but maybe more important than that, the kind of thing that we still are being challenged by, and hopefully some of you might have a better solution than what we apply now. And so I'll talk about several projects all the way from metagenomic deconvolution to copy number variation, and I'll try to show some common theme along this project. And toward the end, if time permits, I'll just show you maybe a few slides of how do I see the challenges and the opportunities ahead in microbiome research. And so to start with, the human microbiome is one of those things that if I gave the same talk 5 years ago, I needed to spend probably 10 slides to explain to everybody what is the human microbiome and to convince you that you have a microbiome. But nowadays, it's actually pretty hard not to know something about the microbiome, and the way that one of my grad students recently put that, microbiome seems to be getting more covers than most B-list celebrities. And that is probably not surprising that we have this fascination with the microbiome, because there is something really intriguing about thinking of humans not as a crown of the evolutionary process, but at least for a microbiologist researcher, more as a talking, walking ecosystem that houses a much more interesting entity, which is a microbiome. And so I will not tell you a lot about the microbiome. I'll just have my microbiome 101 slide just to make sure that we're all on the same page. And so what you probably do need to know about the microbiome, if you did not hear about it before, is that first of all that we are mostly microbes. There are some debate on the exact numbers recently, but at least some estimate argue that there are tenfold more microbial cells in each and every one of us than human cells, although again that has been confronted recently. You should know that the microbiome is complex and diverse. If you just look at your hand, there might be 150 different species on your hand. If you look into your gut, which is a slightly more rich community, you might find close to 1,000 different species, 100 trillion microbial cells. And of course, what makes life interesting and a bit of a nightmare for most bioinformaticians is that it's a highly variable system. So even if you look at your 150 species that you have on your left hand, they're actually totally different than the species that you have on your right hand. The microbiome that I have in my own gut is probably very different than the microbiome that each of you have in your own gut. And those variability is expressed over time in different sites and across individuals. We know that the microbiome is crucial for a lot of host-related processes, all the way from harvesting energy from your diet to protecting you from pathogen to priming your immune system. We know that it is associated with specific diseases, and I'm specifically using the word associated because in many cases we don't know whether it's a causal relationship, although there are some settings where this has been shown quite nicely, but at least what we know is that the composition of the microbiome in terms of which genes are found there, which species are found there, are different and have specific differences in specific diseases in the same way that Thea demonstrated in the agricultural samples. And the reason that we know a lot, because one of the main diseases that have been addressed is obesity. And this is just my very simplistic interpretation of that. And obesity, in fact, is one of the cases where there has been shown a causal effect. If you transfer a microbiome community from an obese individual, from an obese mouse to a lean mice, they actually gain more weight from a singular diet. And so this is one of the poster child of microbiome research. And the reason that we know a lot of that has to do with meta-omic technologies, basically the idea that we can apply the same molecular assays as genomic, transcriptomic, proteomic, and metabolomic, not to an isolated species, but to a whole community of microorganisms, just like the metagenomic example that Thea described. This picture, by the way, is my kid. I gave a public talk about metagenomics, and the only way I thought about exemplifying what it means is to take all his toy animals and put them in a blender. He was not very happy about it, as you can see. But that's the idea of metagenomic, really just applying all those molecular techniques to a mixed community of microorganisms. And I'll talk more about this in a second. And using this kind of techniques, as I said, there have been found multiple associations with diseases, not only obesity, but many, many different diseases, ranging all the way from cardiovascular diseases, neurological disorder, inflammatory bowel diseases, and many, many more. And again, in many cases, the only thing we know is that there is a compositional shift, either in the content of genes in the metagenomes or the composition of species in the community. But in many cases, we don't really know whether it's a cause or an effect. But there's definitely been a lot of research in this front. But what I want to argue now, and I'm assuming that most microbiome researchers would agree with that, is that even though we've been studying the microbiome very intensively for the past 5 or 10 years, most of the fundamental questions really remain unresolved. For example, we don't really understand why we see these differences in the composition of species and the composition of genes. Does it have to do with the interaction between the different species, the host genetics, the environment, your diet, your immune system, historical contingency. We just don't know all that, and probably all the factors that I named are contributing to the composition of the microbiome, but exactly what is the contribution of each factor and how they balance each other is something that we still really do not understand. We also don't really understand how this variation that we consistently see in the composition of the microbiome ultimately translate, if it does, to an effect on the host. Is it important to have more or less Bacteroidetes in your microbiome? Does it important to have fewer Firmicutes species in your gut? We really, in many cases, don't understand that. We also don't understand, in many cases, the link between the composition of different facets of the microbiome. How exactly changing in the ecology of the microbiome translate to changes in the genomics, translate to changes in the metabolomic, and so on and so forth. And if we're thinking about a future of actually designing microbiome manipulation, which is the kind of future that we all hope to see pretty soon, the question is how can we design those kind of interventions? That's something I'll get back to at the end of the talk. And what I want to argue is that some of these gaps in our understanding has nothing to do with the data that we collect, but really with the way that we analyze that. And in order to try to highlight that, I want to go back to the very early days of microbiome research, and I'm not talking about 10 or 15 years ago. I'm actually talking about this guy, Antony van Leeuwenhoek. He's considered by many the father of microbiology. He was an extremely interesting person. He invented what is now known as a van Leeuwenhoek microscope, which is really nothing more than a magnifying glass mounted on a brass plate. He was a very good land grinder, probably had an extremely good eyesight. And he was the first to actually observe the microbiome and microorganism in general. And instead of telling you what he found, I actually just want to read to you from a letter that he wrote to the Royal Society of London in 1683. And so he said, though my teeth are kept usually very clean, we are talking about 17th century hygiene, so not totally clear what it means. Yet when I view them in a magnifying glass, I find growing between them a little white matter as thick as a wetted flour. In this substance, though I could not perceive any motion, I judged there might probably be living creatures. So he assumed that there are microorganisms, of course they were not called this way back then, on his teeth. And then I took some of these flour, and there is a beautiful description that I omitted here about how he prepares a sample. He was actually a very rigorous scientist, and he took rainwater because he assumed they were sterile, and he mixed this in a very specific way. And then to my great surprise, perceived that the aforesaid matter contained very many small living animals which move themselves very strangely. And in fact, he goes on and describes this organism in so detail that we can guess from his drawing exactly which species he found on his teeth back 350 years ago. And then the letter goes on to describe a really beautiful set of comparative analysis. So he compared the bugs that he found in his own teeth to the bugs that he found on kids and on women. There is a really terrific paragraph about looking at the microbiome of a drunk person, which, as he said, he assumed that the constant consumption of tobacco and alcohol killed all those creatures, which of course was not the case. And he concluded his letter with this great statement, which is the number of these animals in the scarf of men's teeth are so many that I believe they exceed the number of men in a kingdom. Right? And as I mentioned before, we now know that he was pretty much on the money. If anything, he underestimated just how diverse and how complex the microbiome is. Now, if we jump now 350 years into current days, and we ask what has really changed from van Leeuwenhoek days to current days, I want to argue that the machinery, of course, changed a lot. So he used this very simple van Leeuwenhoek microscope, and you using a slightly more expensive machinery. He was able to characterize 5 or 10 species, and we're able now to characterize hundreds and thousands of different species and tens of thousands of genes in each samples. But if we think about the kind of analysis that was done, it's actually pretty similar. It was a comparative analysis. He made a list of bugs, and he looked at the list that he found in his own teeth to the list that he found in somebody else, which is very similar to what many microbiome studies are still doing today, of basically cataloging the microbiome, cataloging the species, cataloging the genes, cataloging the transcript, and comparing between 2 sets of samples, between a specific disease cohort and a control, between different time points, and so on and so forth. And again, there are a lot of other studies now that are trying to get more and more into mechanisms, but the vast majority of microbiome studies currently are still very much descriptive and comparative. And why this is not enough, why one kind of van Leeuwenhoek-like analysis is limited, it's first of all because cataloging component is simply not enough. Interactions are important. The analogy that I like to use is like trying to understand what's the difference between an iPhone and an Android by smashing both of them and just counting the number of transistors. right? You need to look at the wiring diagram. You need to think about the interactions. And of course, the other part of that is that nowadays, when we are able to characterize not just the set of species in the microbiome, but also the genes and the transcript and the many other type of omics that we now have, they all represent the same system, but from very different perspective. And we need to think about the way that those different omics relate to one another and interact with one another. And so what is it that we're missing in a single, or actually 2 words? It's systems biology. We need to apply a system thinking in analyzing the microbiome and trying to take into account and to account for those interactions between different components of the microbiome and between different types of data that we're generating. And this is what my lab is hoping to do and what we're trying to promote for the last few years. And we're doing this in several ways. And so the first line of work that we started probably 7 years ago nowadays is to try to model the microbiome. Basically take the standard system biology approach of having a complex system. Let's try to build a computational model of that. This is, of course, not a simple task, and we're not hoping to solve it anytime soon, but we're developing more and more tools to move beyond model of a single organisms that have been developed for the past 30 years to modeling interaction between organisms and modeling a community of organisms. And we have developed a whole array of different methods that will allow you to take genomic data, build models of simple communities, and predict the interaction between species, predict competition between species. We have a model now that is allowing us to predict how the abundance of different species behave in response to different diets. And it's important to know that we are trying to build mechanistic models. It's never about just having a black box that is predictive. It's really about trying to model the mechanistic interaction, in our case, focusing on metabolism. The second line of work is trying to develop different methods that integrate different omic data from the microbiome, again, not based on statistical association. I'll talk about this in a few slides, but based on what we think about mechanism, what we know about ecology and about genomics and about gene activation and metabolism. And then just for completeness, I would say that the last line of work in our lab is simply applying many of these tools to a really cool data set, mostly that we get from collaborators. And those are projects that we became involved in the last 5 or 4 years, where we study everything from the microbiome of children with CF to the microbiome in IBD to the microbiome of killer whales and frogs and chickens and cow and everything. And these are just fun projects to work on. And so for the talk today, I was initially hoping to talk about both of this topic, both the modeling work and the meta-omic work. But then on the way over here, I had a lot of time to think while I was driving and realized that a lot of the modeling work has actually been published several years ago already. And so the tools are there, and you're welcome to use them. And if you have any question, you can definitely talk to me offline. But I really wanted to talk mostly about this Cross-Meta-Omic Analysis, which I think that for a bioinformatic audience is probably could be more interesting. And so why are we even trying to develop a method for cross-omic analysis? Well, if you're following the microbiome research community in the past 2 years, you realize that cataloging the set of species in a community, or even cataloging the set of genes in a community, is no longer enough. And we're seeing more and more attempts to characterize the microbiome using many, many different omic technologies. And so again, it was very common until about 5 years ago to just look at 16S analysis, where we basically look at one gene from each bug and using that as a way to characterize which species are there and what the relative abundance is. Thea described metagenomics, which is trying to take the entire set of genomic information that we have in the community, lyse everything, and get this mixed sample of all the different genes that are there. And as Thea described, in many cases, we get a sense of what functions are there, even though we don't necessarily know which gene came from each species. And more recently, we see more and more studies that are applying the same approach, a meta approach, to analyze the transcriptome of the community, the proteome of the community, and the metabolome of the community. And at the end of the day, we're getting now multiple samples for which we have information about species, gene, transcript, proteins, and metabolites, right? And so, of course, looking at all those different data is great, but what we don't want to do is to be the 7 blind people that are trying to study an elephant and never talking to one another and getting totally different answers, right? So just generating multi-omic data is nice, but what we absolutely need to do is to integrate the different omics, right? We need to figure out how do we use the fact that we have both taxonomic information and genomic information and transcriptomic information. And that's something that, again, is not something that it's not a new concept, and we do see a lot of studies in the past 2 or 3 years that are trying to do that. For example, there have been studies that show that the abundance of genes in the metagenome tend to be highly correlated with the abundance of transcript in the transcriptomes, right? And of course, there are some outliers, but generally, the set of genes that you have in your metagenome are the same set of genes that are being expressed. There are multiple studies now that are trying to correlate the abundance of different species to the concentration of metabolites in each sample. And you see here an example from a study of the vaginal microbiome, where you can see that there are specific taxa that tend to be high in abundance whenever there is a specific set of metabolites that are high in concentration, and you see a different set of taxa that are abundant where other metabolites have high concentration. And you see more and more of these studies, but again, most of these studies are basically using statistical association and correlation. And this is, of course, an important first step to try to understand the structure in the data. But coming from a system biology approach, what I want to argue again is that we're missing a lot by not looking at mechanisms, not looking at the mechanistic interaction between those different types of data. And specifically, if we think about those data, or those omics that I described above, there is almost always some information that we have from genomics, from ecology, from metabolic behavior, from enzymatic control, that will tell us something about what is our expectation in terms of the link between the different omics. For example, if we can use ecological information and information about the way different cells behave in the presence of other organisms, we might be able to link the set of species that you have in your community to the set of transcripts that they actually express. If we look at information about enzymatic activity, we can mechanistically link the abundance of proteins in your samples to the concentration of different metabolites. And what our lab is trying to do is to develop exactly those kinds of methods that take those different omics and try to integrate them, but taking into account this kind of mechanistic information about the different omics. And in the talk today, I will mostly focus actually on the simplest type of data that is possible, which is species composition and gene composition. Again, so now where I know what are the different species and their abundances in the community, and what is the gene families that I find in the metagenome, exactly the same way that Thea described it. And what I want to argue is that what links the two is simply genomics. And let me explain to you exactly what I mean. So let's say I have some microbiome sample, and of course the microbiome sample contains many different species that differ from one another not by their color, but by the set of genes that each one encode, which I mark here as letters, right? And now current analysis can do one of two things. They can try to characterize the set of species in the communities and their abundances using 16S surveys or other kind of surveys to analyze the taxonomy of the community. And you can create this beautiful pie chart that will tell you that half of your community is E. coli, and 30% is Erectale, and another part is Firmicutes, and so on and so forth. And then you can use shotgun metagenomic approach, as we've seen in the previous talk, that will basically last the entire community, totally ignore the species of origin, right? Try to align each read to a specific set of annotated genes, and get a sense of what is the abundances of different gene families or functional pathways in the community, right? And those two kind of analyses are now routinely done almost in any metagenomic study. And what I want to argue is that these two types of profile, the taxonomic profile and the functional profile, are of course not independent. And what links the two is genomics. Because if I actually have information about the set of genes that each species encode, then it's very easy to realize that the abundances of different gene families in the microbiome is nothing more than the genomic content of each species weighted by the relative abundance of the species in the community. Right? And put it as a simple equation, the abundance of each gene family in a sample is basically can be calculated by the copy number of this gene in each genome weighted by the relative abundance of these species in the community. Okay? And again, this is not earth-shattering in any way whatsoever, and every researcher that ever characterized the microbiome probably think about that in the back of their hand, in the back of their mind. What is surprising is how underutilized is this link in the analysis of taxonomic and functional information. And in most cases, the analysis of the taxonomic information, the functional information, is done completely independently. you will see more and more studies that are saying this bug tend to be enriched in this disease, and this functional pathway might be overrepresented, but nothing about why these two data sets look the same. And it turned out that you can do quite a bit with that. And so kind of as a side note, I want to mention that one of the things that actually a few of my students are currently working on is try to visualize those links. And so again, you've seen, if you do any kind of microbiome analysis, you see a lot of these plots. Right, so this is basically the abundance of each phyla in a specific set of samples, and the abundance of different functional families. This is the same kind of plot that Thea showed in her talk. And one of the things that our browser is now trying to do, beyond just allowing you to dig deeper into each of those levels, is to show you the link between the species and the functions in a way that now you can pick a specific taxonomic group, and it will calculate how much of the functional composition that you have in your samples is being contributed by this taxonomic group. Okay, and so this is a nice tool that we hope to release in a couple of months, and it will really allow you to view both of those profiles, but also view the links between them. But of course, as bioinformatician, we want to do something that is a bit more sophisticated. And one of the things that we showed several years ago that you can do is to actually utilize exactly this set of relationship between the taxonomy and the function in the cases we actually do not have information about the genomic content of organisms, or put differently, where you actually are missing reference genome of most of your community members, but instead you have multiple samples. You can use this set of equations to simply solve, giving the taxonomy information and the functional information, solve the copy number of each species in the community for each gene. In other words, what we can do here is deconvolve a metagenomic sample and figure out which gene come from each species in a way that it will explain in the optimal way the link between the set of species that you have in a sample to the set of genes that you find. One way to think about it is if we're trying to basically reconstruct the genomic content of species without actually assembling the species, just by looking at covariation between species and genes in the community. And we show that it works on several different settings. The one example that I will show here is data from the Human Microbiome project. This is a large set of samples from the human body, the gut, but also many other sites. And what you see here, every row here represents a species, every column represents a gene, and whether there is a dot or not represent whether this specific species encode this specific gene in their genome or not. As you can see here, we focus specifically on a clade-specific gene, in other words, genes that are unique to a specific set of species. And the reason we do that is because these are the genes that we usually want to know about. And the black line represents those reference genomes that we have and whether they have a gene or not. And the blue line represents our deconvolved species. In other words, what we predict each genome should contain based only on the abundance of species and the abundance of gene in each sample without taking any information from the reference genome. In other words, even if I did not have any reference genome from this specific clade, just by looking at those convolved samples, by trying to deconvolve the metagenome, I can predict exactly the set of genes that each genome should contain. So this is one approach that you can do, and it was really something that we were excited about. But then we started to talk to some of our clinical collaborators, and it turned out that they're not that interested in that. And the reason for that is because whenever they had a microbiome study, it was always in a comparative setting. In other words, the standard setting was having two sets of samples, cases and controls, again, analyzing the taxonomic profile of each sample, creating those pie charts that I described before, and identifying specific set of taxa that tend to be overrepresented or underrepresented in the disease. They can also do functional profiling using shotgun metagenomic, characterizing the abundance of each gene or each functional pathway in the samples, again, quantifying their abundance, and then identifying specific pathways that are overrepresented or that are more abundant in the disease case. And what they really wanted to know was whether we can link these two. In other words, they didn't care to know how much of the function in a specific sample came from a specific species. What they wanted to know is, can you explain to us why do we see a shift in the disease in the abundance of the function? And so they wanted to ask a question like, if we see a shift in some functional pathway, is that because of just one or two species that change in abundance, or is it because some community-wide dysbiosis, community-wide imbalance in the abundance? Can you actually try to quantify the contribution of each species to the shift that you see in each function? And more generally, if I want to identify the one bug that I need to try to eliminate from the community in order to reverse some functional shift that I see in the disease, can you tell me which bug it is, right? And this is a nice example for a project that when we first thought about it and we talked to our collaborator, we said, yeah, totally, we can do that probably in a month. It turned out that that was a bit more complicated than that, because it turned out that decomposing shifts is much, much harder than decomposing abundances. And so, and in fact, even if you have a perfect decomposition of the abundances of all the different functions in your sample into species-specific information, put differently, if you know exactly how much of each gene in each sample came from which species, figuring out which species contribute to the shift that you see between the two sets is not simple. First of all, there is no trivial mapping between how much you contribute to the abundance of the gene to how much you contribute to the shift. As a simple example, if you have a species that is very abundant in the samples, but is not variable between the disease and control, it will contribute a lot to the abundance of the gene that you see, but it will contribute nothing to the shift. That's not the guy who actually made a specific function be more abundant in the cases. Another problem is that a lot of what I showed you so far relied on this linear, beautiful relationship between taxa and function. And of course, that totally goes out the window when you're talking about shift, because shifts are measured by usually nonlinear metrics. If you're applying a t-test, if you're applying a fold ratio, if you're applying a Wilcoxon test, all those metrics are not linear, and so our assumption about how to measure those actually do not hold. Community context is important. It's not just about whether a specific species changes in abundance, but how is it changing in relation to other species. Covariation between taxa could be important. And just to give you a simple example, if there are two taxa that have a similar set of genes, but they tend to be anti-correlated, in other words, when this guy is going up, the other one is going down, then each one of them seems as if it's contributing a lot to the shift, but combined they contribute nothing because they compensate for one another. And to be a bit maybe more argumentative, I would say that it's also not totally clear how to define a decomposition of a shift. How do you define how much of a t-test score was attributed by one species versus another? And so obviously it did not take us a month. It actually took us about a year. And that was done by Ohad Manor, a very talented postdoc in my lab that took on this project. And he developed this really beautiful pipeline that is called FishTaco, for reasons I cannot expose at this point. But basically, FishTaco is receiving information of the taxonomic profile of each community, the metagenomic functional composition of each community. It can get information about reference genome, if those are available. If they are not, it will predict the genomic content of each species using the deconvolution framework that I described before. And at the end of the process, it will tell you exactly how much of each functional shift that we see is attributed by each species. And again, I will not go into too many details. I'll just mention maybe two principles that we're doing. So the first one is that a lot of what we do is based on permutation analysis. In other words, we're trying to permute the community composition and see how it would affect the functional composition, of course, in silico, because that way we can actually maintain the community context. We can actually ask what would happen if this species would not change in abundance given the covariation with all the other species, right? The other really nice component of this framework is using a framework from game theory that's called the Shapley value analysis. It's named after Lloyd Shapley, which very sadly actually passed away earlier this week. And he developed this beautiful framework that allows you to determine a fair contribution of each player in a multiplayer game. In other words, if you have, for example, if you think about a set of authors that write a paper, and you have some score that is assigned to each paper, so you know how much this group of co-authors is worth, can you tell me what is the fair value of each author by itself? Right? So Shapley developed this a framework that allows you to take all those convoluted information, basically the scores of all the subset, and figure out what is the correct contribution score for each author by its own, we are basically applying the same framework to our species. In other words, we are calculating the contribution of set of species, and by doing that, we are taking into account how different species covary with one another in the community, and then use these scores to assign a specific score for each species on its own. And just to give you a sense of how this analysis, what the kind of result that this analysis allows us to have, this is what happened, for example, if you apply FishTaco to a cohort of type 2 diabetes. So this was a cohort that was published a few years ago, and if you look at the metagenomes of a patient with type 2 diabetes versus patients that are healthy, and you're looking at those specific 3 sugar transport, you find that all of those 3 sugar transport tend to be much more abundant in patients with type 2 diabetes compared to healthy individuals. And again, if you're only using metagenomic analysis, this is where analysis stops. Basically have a functional shift score. As you can see, they are very similar to one another. If you apply FishTaco, you can take this score and actually decompose it and realize that the reason that you see, for example, this shift in the first sugar transport is about 80% contributed by Firmicutes species that are becoming more abundant in the community of type 2 diabetes patients and are encoding this specific sugar transport, and about 10% contributed by Escherichia species, again, that have this gene or have this module in their genomes and are becoming more abundant. One of the things that you might realize is that if I'm actually counting the contribution of all these species, they do not get all the way to the original contribution that I have from the functional data. And the reason for that is that one of the things that we realized in developing this tool is that there are actually multiple ways to contribute to enrichment. And so all the species that you see here are species that are becoming more prevalent in type 2 diabetes patients and encode this gene, and so it's becoming more prevalent. But there is another set of species, including, for example, this Prevotella species, that actually do not encode those genes in their genome and are becoming less abundant in type 2 diabetes. And so if you look, and again, Thea mentioned this before, if you're looking at the relative abundance of these sugar transport in the community, they're actually becoming, you actually see an enrichment because of those species. I also know that there are a few other species that are actually contributing against the shift. In other words, they are attenuating the shift, but there's just not enough of them to reverse the shift all the way. So this is basically translating this very simple functional shift into a taxon-level contribution score for each species in the community. So that was fun, but what we were really excited about is that when we looked at the contribution profile of the 2 other sugar transport, all of them have been reported to be increased in type 2 diabetes, we see totally different profiles. In other words, in the second sugar transport, Escherichia actually seems to contribute to most of the shift that we see, whereas in the third one, it's Bifidobacterium species that are contributing the shift. And this, of course, have a lot of implication to intervention-based therapies, because basically what it tells us is that if we now want to reverse the shift in one sugar transport while not changing the relative abundance of the other sugar transport, we have in most cases a very specific set of taxa that could be used as intervention target and that would affect a very specific function, potentially without affecting other pathways that we have. And so this is just one of the analyses that we have in the paper. This is not yet published. I will say that we've analyzed multiple cohorts, and we see again and again the same signal where different metabolic pathway, different metabolic models have a totally different contribution score, even though they seem to be related to one another. The underlying species that are contributing are still very different. I will say, by the way, that if we are not provided the reference genome and actually predict the reference genome content from the data, our agreement between the metagenomic functional data and our prediction is even becoming stronger, which attests to the fact that there might actually be annotation errors in the reference genome that when we are trying to use our deconvolution approach, we are resolving those. And generally, our score of how much each species contributes to the shift in each function correlate nicely with what would happen if you try to intervene. What happens if you try to take this major species and remove it out of the community? And so that could be a good tool to start thinking about how do we design a specific perturbation, a specific intervention if we're trying to reverse functional shifts. So again, this is currently in review. What's important to note is that FishTaco is already available online. You can download this, you can run it. It's actually pretty easy to run given the data that you have. We've also implemented a web server that will allow you to generate exactly the kind of plot that I showed before, and that is very, again, relatively easy to do, and you're very welcome to use that. There are several labs that are already using that, and if you have any problem with that, just let us know. We're still trying to improve the tool. I want to also mention, and that's actually a project that Thea described briefly in her talk, and this is when this link between genomic data and metagenomic data can be used to try to correct problems in the data. And so Thea described to you how she used this idea of average genome size normalization. I want to tell you a bit about the history of the project. And in fact, this is a project we never wanted to do. But Ohad Manor, the same guy who developed FishTaco, when he just joined the lab, was really concerned about knowing whether his annotation process is working or not. So he developed several pipelines to annotate metagenomic data, and he really did not have any gold standard to compare it against. And so he came up with I think, a smart idea. And he said, look, I'm going to look for genes that are universal and that are a single copy in every genome that was ever sequenced. Right? So every genome that I know have exactly one copy of this gene, and he assumed that the abundance of those genes in any metagenomic sample would be exactly the same. Because the composition of the community doesn't matter. Each genome contributes exactly one copy of this gene as a community. And it turned out when we actually looked at data, and this is from the Human Microbiome project, when he compared the abundance of this universal single copy gene across different types of samples, they're not at all similar in abundance. And in fact, what's even more concerning is that we try to measure the abundance of those genes in different data sets. And so those are from 3 different large-scale data sets of the Human Microbiome. This is the Human Microbiome project, the MetaHit, and another study of type 2 diabetes. And these are all the healthy samples, right? They're clustering beautifully by study. And of course, that's the last thing that you want to see, right? And so he decided he wanted to fix that, and the way he decided to do that is to trying to use those universal single copy genes as a yardstick to figure out what is the right way to normalize the genome. He actually invested quite a bit of effort to figure out why do we have these differences in the first place. And as Thea again mentioned, some of those, or a lot of the signal has to do with the average genome size being different in different communities, but there are also several other factors, including, for example, how closely related the genomes that you have in the community are to reference genome. There are certain genes that are actually harder to identify than others because of GC content and other genomic properties. And so he basically developed a machine learning-based method that is taking the data of those universal single copy genes and trying to reverse or to renormalize the data such that those genes are now equal and hopefully trying to correct everything else. And so, again, this tool is available online. It's called Music. As you can see, if we now apply to the same 3 core that I showed before, it's getting better. It's not being solved completely, and there are probably many other study-related biases, but at least they're getting a bit closer. But I think what is interesting is that when we apply this to try to identify differentially abundant gene in the type 2 diabetes cohort, it turned out that once you renormalize the samples, you are finding those differentially abundant pathways much more easily. In other words, you can find them with fewer samples, and you can find them with a stronger signal. And so this is, again, something that is already available and can be used. And the next project I want to present is an example where this link that I keep on talking about and highlighting and glorifying between genomes and metagenomes and taxonomy actually breaks down. And so from the beginning of the talk, I made this assumption that if you know the taxonomy of your community and if you know the genomic content of each species, you can very accurately predict the metagenome that you'll find. You can very accurately predict the abundance of each gene. But of course, the problem is that if you look at most taxonomic analysis, they will tell you have, again, 30% E. coli, 15% Erectali, but they very rarely tell you which E. coli, which strain of E. coli, which strain of Erectali. And of course, we know that strain could vary dramatically in the gene content, in the copy number of each gene. And for years, there was this assumption that there is some strain variability in the metagenome. There have been studies that show that there is a lot of SNP that you can find if you're trying to align things to the same genome. But we are interested in functional strain level variation in cases where one genome of certain species in a certain sample has a gene, and in another sample you have a strain that are totally missing this gene. or in one case that it has one copy, in another sample it has multiple copies. And again, everybody assumed that there is a high level of variation, but it was hard to characterize how much because, of course, the last thing you want to do is to start in isolating each and every species from each and every sample and sequence that and trying to detect this variability. And so Sharon Greenblum, a talented graduate student from my lab, tried to see if she can identify how much copy number variation, how much genome content variation there is between different strains in the microbiome directly from metagenomic data. And I always like to show this picture, first of all, because I'm really proud of this specific study, but also because to me it's a great example of what happened if you ask one of your friends who happened to be a fashion designer to design a cover for Cell. And so I tend to say that this is the most Vogue-like cover that Cell ever had. And so basically Sharon developed this pipeline that is relying on multiple factors. It is aligning multiple genomes to create pan-genomes. It's aligning individual reads to those cluster genome. It is aligning the gene families to one another in order to be able to characterize them. It is using those kind of single copy genes in order to normalize the abundance within each genome to the right copy number, and that basically pipeline represent a year of Sharon life. She ran this on an extremely large scale. At the end of the project, she aligned 2.4 billion reads, and I think this was before the revision, where we of course asked to align a different data set, which probably double that. A few hundred genomes and more than 100 samples, and at the end of the process, she was basically able to characterize the copy number of 40,000 genes. Or put differently, she now had an estimate for the copy number of each gene in each genome in each sample. And she wanted to use this data to try to estimate the amount of copy number variation. And one of the decisions that she made, which I think turned out to be really a smart thing to do, is rather than looking at those estimated copy number and going back towards the reference genome and ask, is it different than what I know about the reference genome, is to actually compare the copy number between the samples themselves. And by doing that, she's totally bypassing the problem of wrong annotation in the genome, of genomes that are not covered correctly, or genomes that have very poor coverage. And by doing that, she was able to discover quite a bit of copy number variations. So that's, again, to give you a taste of the kind of data that she has provided. This is, for example, the copy number variation. And so what you see here on the bottom are different samples. Those are different genes. The gene that you see here on the top are those marker genes, those universal single copy genes. And as you can see here, the box size and color represent the estimated copy number in one specific species. Right? So this is not in the metagenome. These are estimated copy numbers specifically in this Dysbiosis. Right? And as obviously expected, those marker genes tend to be constant in copy number. But if you look at the set of transfer genes, they show incredible copy number variation between samples, whereas in some samples, some genes have a copy of 1, and in other, there's a copy of 15. Right? There are some cases where we can actually show that this copy number variation is correlated with the host of this, with the state of the host. For example, this specific MFS transporter in this specific species tend to have a much higher copy number if you are trying to estimate in sample from patient with inflammatory bowel disease compared to healthy samples or obese samples. Right? And by the way, the nice thing about this example is that it has been known that this gene, in general, is more abundant in the metagenome of a patient with IBD. What we show here is that this is not only because those species are more abundant, but because the copy number in each genome of these species is actually higher. To give you a sense of the scale of the data that she generated, what you see here is basically a summary of all the variation that she identified. On the different column represent different gene families, or KO. Those are the different species that she has characterized. Each colored bar represents a gene in a species that was found to vary dramatically between the different samples, and as you can see, there are a lot of them. But probably the most important take-home message is this bar plot on the side here, where it's basically listing the percentage of genes in each species that tend to be highly variable between samples. And as you can see, there are some species where actually a quarter of their gene tend to be very highly variable between the different samples. And so next time you see a metagenomic sample and you see some taxonomy profile that tell you you have this specific genome, in fact, or this specific species, in fact, it could be up to 25% of the genes might or might not be there, or might be very different in copy number between those different species. And so the last project, and this is again just another example of the data, just to show the scale of variation that we identify. Each circle here, each Venn diagram represents a species. The gray part represents a set of all the genes in the species. The pink one represents those genes that we know have copy number variation based on reference genomes that have been sequenced. And the darker red color represents those genes that we identify as varying in copy number between the different samples. And you can see in most cases, we do identify a large portion of those variable genes in our sample, and we don't expect to see all of them because not all this variability actually exists in the sample that you're studying. But we do identify many new genes that were never identified before based on reference genes. Okay, and so this method is both agreeing with some variation that that was already characterized, but also revealing a tremendous amount of novel copy number variation between different strains that you have in your microbiome. And so the last project I want to talk about is really just to show an example of going beyond this link between genome and metagenome, and showing you an example. We're trying to start to tackle those other parts of the link between the different omics. And specifically, this is a project that was carried out by Cecilia, another graduate student in the lab, that was just actually published a couple of months ago. When we tried to ask, can she actually use this same approach of using mechanisms, not only to link the genomes or the species to the metagenome, but all the way to the phenotype of the community, which is the metabolome? Can she actually explain, using information about the ecology of the community, which species are there, the specific differences, whoops, not supposed to happen, the differences that we see in the concentration of different metabolites in the sample. Right? And again, as I showed before, there have been multiple studies that have tried to do that based on correlation, finding specific species that when they're high in abundance tend to be correlated with specific metabolites that are high in concentration. But she wanted to try to do this link mechanistically. And in other words, she was hoping to be able to take the taxonomic information, to use genomic information to predict actually the content of the metagenome, to take this metagenome data or predicted metagenome data and use a metabolic model to try to predict the set of reaction and the set of metabolites that can be synthesized and degrade, and then to use metabolic modeling to try to predict the concentration of different metabolites. Okay? And this is basically the pipeline that she developed. Again, it's taking multiple samples, predicting the gene content, using metabolic information to predict the metabolic potential, basically how good do we think each community is in synthesizing and degrading specific metabolites, using that to predict the abundance of metabolites between the different samples, and then compare this to actual metabolomic data that we had. And I have to say that when we started this project, we basically assumed it would never work. Right? Because if you think about the number of assumptions that we do on the way, we assume that the genomic information that we have from reference genome really represent the specific species that we have in our community, when just one project ago I show you that we know this is not the case. We assume that the abundance of the gene in the metagenome is a good representation of the abundance of transcript in the metagenome. We assume that the enzymatic rate do not matter. We basically were trying just to link the ecology directly to the metabolome, making those multiple assumptions about the mechanism. And so again, we started this project assuming that we may be able to predict 1 or 2 metabolite abundances. When Cecilia actually applied this to a set of samples that we have from the Father of Microbiology, for which we have both information about the taxonomy and metabolomic information, it turned out that on average she was able to predict variation, to explain mechanistically variation in about 1/3 of the metabolite that were measured. Right? And so that really showed us that even though we're making all those simplifying assumptions, the composition of the ecology, giving mechanism, can tell us a lot about the variation that we see in the metabolome without trying to fit the data, without trying to learn the parameters. One of the interesting things that we discovered is that in addition to this 1/3 of metabolite that we're actually being able to explain by the composition of the community, there was another 1/3 of the metabolite that it's not that we were not able to explain them, but actually every time that we were trying to explain the variation, we're reversely correlated with the actual measurements. In other words, there are a certain set of metabolites that when we predict the community can synthesize them, there is less of them, and when we predict the community can degrade them, there is more of them. Right? And this is something that we're still trying to understand. Our best guess so far is that this basically represents the difference between metabolites that are being controlled by the microbiome, so metabolites that are being synthesized by the microbiome, versus metabolites that are controlled environmentally and are selecting for the community. In other words, those metabolites that whenever they're high, the degraders of those metabolites also tend to be high, are those metabolites that are selecting for the higher abundance of species that are able to use those metabolites as energy sources. And in fact, this is something I don't have on the slide here, but we recently analyzed microbiome samples for which we have taxonomy information, metabolomic information, and dietary information. And we show that this really nicely correlated. And so those metabolites that we tend to explain well are the ones that are not found in the diet. And the ones that we tend to anti-predict, if you will, are exactly those metabolites that are present in the diet and likely selecting from the community. I will just also say that one of the nice things about this approach is that it is allowing us not only to explain the metabolite that we find, but also because we have a mechanistic model, we can say, for example, which species is contributing to this agreement. And one of the things that we found, for example, in the vaginal microbiome is that those species that actually explain the variation in metabolites were not necessarily those famous bacterial vaginosis-related species, or those species that people that study this system assume are the contributing to variation. And so I think I'll almost end with that. I'll just want to share with you a few take-home messages that I think are important. And so first of all, I do believe that system-based integration is crucial. Right? You can find correlation, you can find association. That's a really valid and important way to start characterizing data. But if you want to get to a mechanistic understanding, you have to introduce those kind of system-level consideration and mechanistic links into your model. You have to utilize information about molecular interactions, genomics interaction or genomic dependencies, and ecology. And that new tools are available. And so again, we mentioned several tools that are already available. Everything that we develop is, of course, publicly available, and we're trying to make it as easily accessible as possible. And if there are any concern, you can always contact us. A few comments that I always have to note whenever I talk about models or model-based method is that we know that those models are problematic. I like to quote Arthur Rodenbus, a philosopher of science, that said that the best model for a cat is another cat, preferably the same cat. It is worth noting that he said this in irony, right? In other words, you cannot use the same cat to model your own cat, because if the model system is as complex as the system that you're trying to model, you cannot learn anything new from that. But probably the simpler version of that is the one that was stated by George Box, that all models are wrong, but some are useful. And I think that we know for a fact that all the models that we use are too simplistic. We're making a lot of simplifying assumptions. They're obviously incomplete, but I think they're useful to study interesting things about the microbiome. I just want to show you a few slides about maybe future challenges, and I'll be very quick about that. And so first of all, it should be clear that the vast majority of the work is still ahead. Everything I show you today is really trying to work on this link over here between species composition and metagenome. And we're starting now to work about trying to link the metagenome to the metabolome. All those other links are still very much have not been utilized in studying microbiome data. There are a lot of problems that need to be addressed. How do we take into account the environment? A lot of the modeling that we do and a lot of the methods that we develop are kind of assuming that the microbiome is living in this ideal chemostat environment where, in fact, the host metabolism and the host immune system can have a major effect. A question of scale and resolution are crucial. What resolution do we need to characterize a microbiome in order to be predictive? Do we really need to take into account strain level variation, or it's enough to just look at the genus level characterization? What temporal and spatial scale do we need to look at? Is it, if you're trying to study, for example, some temporal variability, do you need to sample your microbiome every day, every hour, every month? Do you need to take into account heterogeneity in the way those bugs are organizing in space or not? A big problem that I always have in every talk is this functional gray matter. And so you all heard about the concept of microbial dark matter as those bugs that we have not yet sequenced. Functional gray matter is a term that one of my students coined as a way to represent those genes that we have sequenced, but we know nothing about their function. Right? There's this huge problem of annotation of, and again, I think Thea showed this in her talk, of having this half of the gene being as not annotated or not really aligned because we actually don't know what they do. And so this is always a limiting factor to everything that we can do. it's important to know that everything that we do in our lab, and I presented everything as if our ultimate goal is really to understand the microbiome, but of course, what we really want to do is something much more noble than that. And so I'm assuming many of you have seen this beautiful cartoon about going to the doctor, and the doctor saying there's something wrong with your microbiome, and you wait a minute, and then they come back with a wolf and said, do you need water for that? And I have to admit that we have no problem whatsoever using wolves as microbiome-based therapy. The problem we have is with this part, because it's totally missing the fact that we need to be able to figure out whether you need a wolf or not, or what strain of wolf. In other words, what's currently missing from our attempts to design or to develop microbiome-based therapy is rational design. How do we know which species we're trying to replace? How do we know which species are causing a specific shift? And some of the methods that I showed before, I think I alluded to what we hope is their application for design. I just want to maybe mention very quickly this one project that was just published earlier this week, where we're actually trying to address this problem head on. And one of my students tried to ask, can I design an algorithm that is giving a specific set of species, a specific set of available metabolite, and some set of desired metabolite, identifies the set of species that I need in order to catalyze or to synthesize, depending exactly what you're trying to do, from the available nutrients to the desired metabolite. And again, this is a very preliminary attempt to do that, and that could maybe be used as a first filtering stage, but this is the kind of thing that I hope we'll do more and more in the future. And so I think I'll end here. I'll just want to thank everybody that actually did the work. I think I mentioned most of them by name, but just to make sure, this is Sharon that worked on the copy number variation, Ohad over here that worked on FishTaco and music. I think there are a few other, Alex that has developed the design framework, Cecilia that is linking the taxonomy of the community to the metabolome. I also always like to note to the fact that this is my lab and they're always looking into the future, but everybody's looking into a different direction, which I'm not exactly sure what it means. Some of our collaborators and some of our sources, this is how we actually look in the lab. And the last thing I want to note is that, I started with the fact that we are a computational lab and we are bioinformaticians, but we do have one experiment that we do once a year. This is our annual Fermentafest event, when we're basically trying to ferment every food that we can find and eat our own model system. We did not kill anybody yet, but I think we're getting close. And I'll be happy to take questions.
When you're doing these deconvolutions, like I guess in particular I'm thinking of the first one you showed, but kind of in general, you're kind of at the mercy of whether the system that you're trying to invert, I guess, is well defined or is well conditioned, right? So I guess two questions. One, has that ever come up?
So what do you mean by well conditioned in this case?
Well, like if you're trying to sort of regress, you're trying to go from the product to the sort of cause by linear regression, right? You're somewhere in there you're trying to invert a matrix, right? And you can only do that if it's well conditioned, or right? And so I'm just wondering, A, sort of has that ever come up, and B, what does your uncertainty look like? Like what kind of how do you sort of report confidence in these regressions?
So the question was, in the deconvolution work, how do we deal with cases where the matrix that you're basically trying to regress is not well conditioned, and can we report confidence? And this is definitely a big problem. I have to say that a much more immediate problem is that this specific method is relying on having enough samples. And ideally, you want to have samples that are on the order of the number of taxa that you try to deconvolve. And that's in most cases is a real problem, specifically trying to deconvolve individual species. One of the things that we do in these scenarios is trying to deconvolve a higher taxonomic level. So in the HMP example that I showed, we're actually not deconvolving individual genome, we are deconvolving whole genuses. Right? And that, of course, has some problems because the assumption that is underlying this entire model is that the genome in each species, in each sample is the same. And of course, when looking at genus-level decomposition, this is not really true because it depends on the relative abundance of different species within this genus. It is a problem, and I don't view that as a method to actually deconvolve community or to obtain genomes by itself. We view that as an additional tool to help you, for example, bin samples before you try to assemble. There are now, since we published that, there have been multiple other techniques to try to do that. Probably the one that got a lot of attention is using high-C data to trying to identify reads that are physical, that have physical proximity in order to identify a group of reads that actually come from the same species. Those have been pretty successful, and I think we'll see more and more of that in the future, and some of that came from our department and from our collaborators. As far as confidence is concerned, that's a real problem, right? And in most cases, I could probably provide a confidence interval or some sort of significance score if I would have enough samples to use as a test for our deconvolution method. There are a lot of other concerns. This is a place where I like to quote my PhD advisor that said the man got to make a living somehow. In other words, we know there are a lot of assumptions and we know there are a lot of factors that will not work, but as an approach, we still think it's a valid approach. Okay. Yeah.
So this is maybe not a well-posed question, but I'll try anyway. So you mentioned early on the idea that looking at components is like breaking two columns, looking at components, ignoring interactions. And so in this context, to what extent are the strains and species interacting, and how does that modulate their concentrations of their genes? Can you put a number on how interactive the components are, how connected?
So first of all, I wanted just to mention that when I talk about interaction, it's true at multiple levels. It's not only interaction between species, it's also interaction between pathways and between genes. Coming back to the question of interaction between species, this is actually something we worked on quite a bit in our modeling work. And one of the things that we've done is develop a framework that allows you to take genome data, translate that into genome scale metabolic network, and predict the level of competition. And one of the things that we've done is then ask if we are applying this metric to the gut and we compare this with co-occurrence data. Do you find that species that tend to co-occur in the gut are the ones that are competing with one another or the ones that are actually cooperating with one another? And we started this project with a very clear assumption that we'll obviously see kind of the traditional ecological approach, which is you see species that have high synergy between them. You'll see species that tend to have cross-fitting between the two species. And what we found was exactly opposite of that. We found that species mostly tend to occur with the strongest competitors. Right? We've done several other projects that have not yet been published where we try to test this on a larger scale. Specifically, we're trying to ask if you're looking across many different metagenomic samples, not only from the gut, but from soil and sea and other cases. And you're trying to find set of genes that co-occur in the samples because different species encode those genes. And again, we thought in the beginning that we'll find this traditional example of one species encoding the beginning of a pathway and the second species encoding the second half of the pathway. We never found that. The only thing we found is cases where species are trying to partition the niche so that one species is using one carbon source and the other one is using another carbon source. All that is to say that if you ask at least what I believe now is I believe that those kind of really fun syntrophic system that everybody is studying, where you have two species that have a very tight cooperation between them, are interesting but are rare. And there are exceptions to the rule. And in most cases where you have microbial systems, most of the species will either compete for the same sort of nutrient or will try to partition the niche so that they will not actually step on each other's toes. And to me, that again has something that I think is important, because one of the reasons that we did this study in the first place, again, is thinking about design of intervention, is wanting to know, should we care, if we want to introduce a specific bug to a system, should we care about which other species are there? And our initial thought that that would be extremely important, my gut feeling now, no pun intended, is that it's probably less important that people assume. Is that answered?
I think you did. are you saying that basically if you look at the expression levels, they would be down-regulated compared to the control where they don't interpret?
So all that is based on genomic data. There was no transcriptome data when we did this project was available. So the only thing I'm saying is that if you look at which genes are encoded by species that tend to share the same environment, in most cases, those are genes that are trying to partition the niche rather than to have cross-fitting between them. Okay, whether those pathways are being expressed or not, that's a great question, which I don't have a good answer for.
Yeah. You showed your the product of counts of species times counts of genes in species equals counts of genes. You had counts of species done with 16S sequencing and counts of genes done with short-read sequencing. Is that difficult? Do you see people still doing both 16S sequencing and short-read sequencing on the same sample? I feel like that's like canvas and way.
So we still see that. So if you would ask me 3 years ago whether 16S sequencing gonna still be something that many people use, I would probably say no. I mean, everybody's gonna use shotgun. It's becoming relatively cheap, and it's easy to get taxonomic information for. And we definitely analyzed some sample where the species information was obtained from the shotgun data using a platform like Metaflan that was developed by Curtis Houdenhauer. What we do see now is this second wave of labs that are becoming interested in microbiome and are trying to look for the cheapest entry point. And in most cases, this is 16S-based analysis. It is correct to say that in most of these cases, they only do 16S, and if anything, they would do shotgun metagenomic on a smaller set of samples. And so we can still use that. Another thing that we can do in those scenarios, and actually something that we've done quite often, is that specifically for FishTaco, where we want to understand the functional shifts, we're actually using this equation to predict the metagenome. Right? So basically using 16S data plus reference genomes, and in the gut at least, we have a good coverage of reference genome already to predict the metagenome and then try to understand the shift that we find there. This was a very long answer to a very simple question, but simple put, we do see this example. There are two that are becoming more rare. Yeah.
Since the human gut is so hard to study, would it be easier to apply all this in bioreactor, the human gut, where you can control intake?
So we never tried to work in a bioreactor data, mostly because we didn't have a collaborator that was interested in that, and it's easier to get gut microbiome data. I would say that a lot of the methods that I presented are at least partly relying on having good coverage of genomes, which is true for the gut, but is totally wrong for marine samples, for example, or agricultural sample, or any other kind of sample. I think that we're getting there, but currently the gut is so well characterized that we're really focusing on this environment. And again, if you have some other type of sample that you're working on and you're interested in one of those methods, I'll be happy to talk to you. In some cases, the coverage of genome will be a big problem.
We'll finish here. Let's thank Dr. Borenstein.
So I just want to mention that this is our last session for the season. We haven't picked next season's speakers yet, so if you have any suggestions for someone you wanted to see that we bring to Vancouver, go to our website, and there's a speaker nomination form that you can fill out, and we'll be evaluating that in the next month or so. And also, I'm the one standing up here, but Van Bach is actually a collaborative effort of quite a few individuals here. So please give them a thank you as well. I'm happy to say that we'll have 2 new people joining us for next season, but if you're interested to volunteer, we're always looking for people to help as well. All right, pizza's getting cold, so.
Yeah.